Welcome to Click for IB Biology. Here we're looking at autotrophs, the organisms at the beginning of food chains and food webs. The two styles of autotrophs we'll consider are photoautotrophs and chemoautotrophs. There's a blank PDF of this presentation down in the description that you can annotate with your own notes. You'll also find there 10 questions that you can do along with the answers to secure your own learning. So let's begin with the photoautotrophs carrying out the process of photosynthesis, and of course, as the prefix implies, using light, photo, in order to construct biomass. In the process of doing so, the light energy absorbed from the sun by the chlorophyll systems of plants and the process of photosynthesis is used to fix carbon dioxide into the biological molecules. The molecules fixed and created by photosynthesis are not just carbohydrates, but also by way of the absorption of nitrates. It's possible to synthesize amino acids for proteins, and the absorption of phosphates allows the synthesis of photolipids that we might associate primarily with our cell membranes and the structure of DNA. So these are the photoautotrophs that we commonly associate with terrestrial photosystems, but also at the margins of our oceans, where it's possible for light to be absorbed in the photic zone of our oceans. However, deeper in our oceans, it turns out that there is another system discovered in the late 20th century, which we now call chemoautotrophic organisms. And from this distribution map here, you can see that these chemoautotrophs are located on the volcanic regions, the volcanic active regions around the world. Deep in the ocean, it was discovered that there were these so-called deep water hydrothermal vents in which carbon dioxide dissolved in ocean waters or extracted from hydrocarbons were fixed into biomass, biological molecules as well. In order to do so, it's necessary to have forms of energy rather than light, and these include things like hydrogen sulfide gas, ammonia, or some of the ferrous ions. If we compare those two processes in this oxidation reduction diagram, here we have the photoautotrophs we associate with terrestrial biomes and also the ocean margins, in which light energy from the sun is fixed by the molecule chlorophyll, and it's turning that energy into another form of energy, which of course is chemical energy. That chemical energy is now used for the reduction of carbon dioxide into molecules such as chlorophyll, and this is associated with the chloroplast and with the Calvin cycle. Contrastingly, in the deep ocean hydrothermal vents, we have chemoautotrophs. These are bacterial species which are able to use things like this ferrous ion and bring about its oxidation, and with that oxidation, the energy here is transferred to carry out the reduction of carbon dioxide, again in biochemical pathways which are remarkably similar in to the Calvin cycle. Indeed, they are the Calvin cycle. Some authors, some researchers believe that the chemoautotrophs probably reflect the original form of molecule formation, particularly through the studies of ribulose bisphosphate carboxylase and its ancient, apparently ancient history in living things. So two different ways in which energy can be fixed into our producers and which initiate, therefore, the transfer of energy from one trophic level to the other. This is the link. Our producers, our autotrophs, in these two styles here, are our link to the energy forms available in the abiotic environment. The next video considers heterotrophic organisms. Here's a chance to subscribe to our channel and indeed the playlist for this particular topic.